Welcome back. My name is Wei Li. I'm a senior investigator at the National Eye Institute. It's a great honor to be the co-chair of this neurobiology session, packed with exciting talks. So we'll get to it. Just quick note, we'll be I got the order from Hong and Jie that we should be very strict on time. So I'll give a kind of a signal at maybe 13 minutes. So you could wrap up speakers, please, at 15 minutes or so. And we will strictly cut it off at 20 minutes. So I have my favorite kitchen tools here at the 20 minutes. So let's get right to it. First speaker, Dr. Zuhang Shen, one of the senior, if not the most senior member of the neuroscience community at NIH. Great works many years from Zuhang, and today he's going to talk about exciting topic on mitochondria and energy and axons regeneration. Zuhang, stage is yours.
Thank you. Thank you, organizer, for inviting me for this fantastic program. I'm happy to share our research, and particularly focusing the neuronal energy research with the Chinese Society of the Biomedical Research and in this area. So when we studied its brain functions, and most of the people focusing on the structure for synapse, axon, or nerve cell or circuit, and also study function about channels, synapse transmission, and learning and memories. But our laboratory is focusing on the energy, which is important for brain to maintain structure or maintain and all the functions. So today timing is limited. I only talk about how energy can support the regeneration after injuries. So, and if anyone interested for the energy, we just have 2 weeks ago, we have the new review article discussed about brain energy. We know that in cortical neuron in human brain, utilize up to 4.7 billion ATP molecule per second to support and drive biological functions. So majority energy is 93% in neuron is come from mitochondria. So in neurons also have another layer of the complexity because it's morphology. So neuron has a very relatively small cell body along axonal process. This is a particular for dopaminergic neuron. You can see in the axon branch, very complex. This is important for the location of energy source. So neuron face exceptional challenge to maintain energy metabolism in the distal axon synapses or glucose. And the mitochondria in axon is highly dynamic. They transport around axon process. They can transport bidirectionally. They can pulse or change directions. They even can, station mitochondria can be remobilized or redistributed. This is a motility patterns is achieved by set of protein complex, which is kinesin adenine modal protein driving the microtubule-based transport, or adapted protein link modal protein to the organelle, and along with anchoring protein to stop mitochondrial transport. So all these proteins, they work together in response to change in neuronal activity or energy or metabolic status. So today I'm focusing on the anchoring protein, which is stop mitochondrial transport. So about 12 years ago, my laboratory isolated protein we named synthophyllin, which is the axon mitochondrial surface protein. So here I show you 3 levels of imaging. So it shows this is localization of synthophyllin. So hippocampal culture neuron with a confocal microscope to show synthophyllin as very small vesicular structure, mainly distributed in the axonal process. Then 3 years ago, in our neuron paper, we're using immunogo anti-synthophyllin antibody to show nicely the antibody label the surface of axon mitochondria. Then we're using STAT super resolution imaging to show single axon mitochondria. You can see synthophyllin on the surface of the mitochondria. So to understand the functions, we make a gene knockout. In the white type hippocampal neuron, it's about 30% mitochondria in the move process axons. But in knockout neuron, you can see almost 80% of mitochondria is moving across the exon process. So this is in vitro condition. Then we confirm this in vivo using mouse static nerve. This is exon bundle. In the wild type mice, you can see about 30% of mitochondria is moving across the exon bundle. In knockout mice, you see more mitochondria, about 70%, and moving across exon bundles. So this suggests the syntrophin, which is to stop mitochondrial movement, which in interaction with the motor protein, which is driving mitochondrial transport, moving and stopping mitochondria depending on the action balance. So it's a reason that optogenetic study to show that selective recruit syntrophin to the multi-mitochondria can stop mitochondrial movement. Vice versa, for recruit motor protein to the stationary mitochondria can recruit stationary mitochondria to multi one. So this is balanced by motility is balanced by the motor protein or anchoring proteins. So now we're back to the nerve regeneration, central nervous system regeneration. So we know this is a major medical problem, is central nervous system, particularly for the aging and mature neuron, lost their regrowth capacity after injury. Regeneration involves many off-cellular processes. So this other cellular process for recovering or repairing require high level of energy consumption. And injury also damages local mitochondria and leading the energy crisis in the locally. And syntrophin, which is stop mitochondrial transport, is highly expressed in the aging and mature neuron. They stop mitochondrial movement, including those damaged mitochondria. So based on this phenomenon, we raised questions whether syntrophin expression in mature central nervous system neuron can contribute to decline regeneration capacity. To address these questions, we make a working hypothesis. So we think syntrophin knockout mice enhanced mitochondrial transport is necessary to replace damaged mitochondria with healthy one, then rescue energy crisis and facilitate regeneration. So here I just show you two stories in the published work and mainly contributed by Bing Zhou and Qi Han. They both running their own laboratory, one in the Beijing, one in the Shanghai. So this is an in vitro study or in the spinal mouse spinal cord injury models. The in vitro studies, we culture cortical neuron in the microfluid chamber systems in the somodendritic area. Only exons can regrow passing through the micrograph into exon terminal chamber. Then Bing can using laser microsurgery to cut exons in this exon terminal chamber. They look for the regeneration. So first we did is a live imaging to show how mitochondrial delivery from cell body to the exon terminal through this exon bundle area. So you see that 1 hour after injury, Y-type knockout neuron, you see mitochondrial transport is more dynamic in the knockout neuron. More mitochondria is moving through cell body to the exon terminal in the knockout neuron when compared to Y-type. Then you can look for 3 days after injury for regrowth exons. You can, by your eye, you can see the more exons regrowth after injury in knockout neuron when compared to Y-type. Then being working on the in vivo using static nerve, we know the mitochondria is very dynamic transport in the knockout mice through exon bundle as static nerve. Then using same after nerve tissues, we do crunch injury. So you can see in the knockout mice, more exons regrowth after injury, the growth even longer compared to Y-type mice. So now it's a challenge to understand the mechanism. Why is transport is matter? So we look for the exon mitochondria in the microfluid chamber systems. We label the mitochondria with GFP mito is green, and TMRE is a membrane potential that only label polarized mitochondria. So healthy mitochondria, suppose its color is yellow, can label by both a dye. But mitochondria lost membrane potential, they lost the TMRE staining, they change the color from yellow to the green. So I showed you a video, and before injury, majority of mitochondria is yellow. But after injury, only 15 seconds, you can see is a mitochondria next to the injury site, they can change the color quickly from yellow to green. That means they lost membrane potentials. So now question is, if we enhancing mitochondrial transport can replace this damaged mitochondria. So this compared to wild type knockout neuron before injury, so all the mitochondria can label by both color, the GFP, mito, and TMRE. But after injury, you can see 5 hours after injury, wild type neuron, but majority of mitochondria, they lost membrane potentials. But in knockout neuron, majority of mitochondria retain membrane potential. That suggests enhancing mitochondrial transport can replace damaged mitochondria. Then we look for the ATP, the energy. So we're using two different ATP sensors. First is the ATP probe called Pulsable, which look for the ATP tips in the growing tips after injury, can compare to wild type. Knockout neurons to show more of ATP accumulate in the growing tips. So we should remember the growing tips require a lot of energy to support neural regrowth. Then we look for is another sensor. It's called flattish-based Redshift ATP probe called GO18-2. So this look for all the cytosolic ATP level in the axon terminal. Then look for the wild type knockout before injury to have similar level ATP, but in the wild type neuron, so after injury, you see is the progressive decline of ATP level. But knockout neuron, you see ATP remains stable. So this suggests enhancing mitochondrial transport can increase ATP, the injured axons. So now we move to this more challenging model systems in vivo in mouse models by spinal cord injury, because spinal cord axon is the longest axon in the mice. So we collaborated with Xiaoming Xu's group in Indiana, and Xiaoming is expert for the neurosurgery for the mouse spinal cord injury. So we choose cervical 5 dorsal hand sections and after 8 weeks injury, then we injection BDA dye, which can trace as an axon, which injection site from sensor motor cortex here. The dye will go into the spinal cord. They label all the new glowing axons. This is a lesion site. Then we look for is the single section staining. Okay, see, this is a lesion site here. And in the wild type of mice, axon is a retraction and dying back. This is very typical for the central nervous system injury. But in knockout mice, axon can passing through lesion site. This is from single section. Then we more challenging to want to make all the whole of the spinal cord, make a 40 section. Then each section after staining, we combine all together by reconstruction together. So you can see each color means one sections. In the wild type of mice, you can see after injury, only a few axons can regrow passing lesion site. But in symptomatic knockout mice, the enhancing mitochondrial transport, you can see hundreds of thousands axons can regrow passing lesion site. So now we can confirm about is mitochondrial delivery. So we did a staining as an injection mitochondrial membrane potential dye and CMTMI injection from sensory motor cortex. Okay, this dye will go into the spinal cord, then going to the regeneration axons. Then we collect the axon in this area, just before the injury site. So here I show you is the 20 axons from wild type knockout. This is a color-coded heat map. So each bar code indicate one mitochondrion. So color means membrane potential. It's a red and yellow, so means high membrane potential, and blue code means lost membrane potential. So by your eye, you can
13
see,
minutes, Suhang.
You can appreciate is in the knockout mice after enhancing transport, more healthy mitochondrial delivery into the injured axon when compared to the wild type of mice. So this is a regeneration, and so is the gene manipulation. And the recent studies we published 2 months ago. So Ning Huang is postdoc, and he just joined the lab and joined the faculty in Xi'an Zhaodai University. So he set up the axon energy battery after neuronal injury. And he found out the injury ischemia can activate an energetic AKT-PAC5 signaling axis within local axons. So this signaling pathway can turn off the centrophilic anchoring function by phosphorylation, which very quickly, effective way. Then turn off the remobilized mitochondria, remove damaged mitochondria from distal axon, and deliver healthy mitochondria to the axons, can replace the mitochondria. So this way can quickly boost axon energy recovery for support regeneration. So this is the imaging show is that after reprogram, is more axons regrowth after injury compared to control neuron, which is showed the much less after regeneration. So another study which is published a few months ago in neurons, we found is the axon energy can also maintained by oligodendrocyte cell. We know oligodendrocyte cell serves the myelination cell surrounding about 97% axon surface of central nervous system. So oligodendrocyte cell can increase axon energy level by deacetylation of key mitochondrial proteins through the NAD-dependent SO2, which is a molecule is absent in neuron, but highly expressed in mature oligodendrocyte cell and released with exosome. So exosome can be uptaken by the axon, can deliver it to the mitochondria to enhance mitochondrial energy levels. So now I just have take-home message. So regeneration requires the recovery structure function, but also have to recover the energy supply, because injury can damage local mitochondria, cause energy crisis, and regrowth program require high level of energy consumptions. So neuron, mature neuron can regain regeneration capacity by enhancing mitochondrial transport and reverse the energy deficit. So after our study published and 2 journals, they have editorial and articles discuss about how mitochondria is so important to make nerve growth and mitochondrial motility facilitate neuronal recovery. This is the whole lab photo, and unfortunately we don't have the photo took by after following in the things of the pandemic. So this is before the pandemic photo, and I might not have able to answer questions. Thank you.
Thank you, Zuhao. That's a wonderful talk. And we have questions from first Hong. Yeah, I guess so. He's asking, what's the benefit of stop mitochondrial movement in the wild type? Why do you need to anchor mitochondria along the exons?
Well, this is very good questions. And actually, we have another study I don't have time to talk about, is that synapses require a lot of energy. And in mature neuron, majority mitochondria, which is a stop in the synapse to maintain and synaptic transmission. And in mature neuron, that require more in developing neuron, when neuron in growth or regeneration, to require delivery, high delivery mitochondria to the growing tips. But after neuron mature, they have stopped in the synapses to support synaptic transmission.
But along the axon, are they aggregates around the node of Ranvier?
Yes.
Is there any?
Yes. They support and a lot of channel functions. So they are calcium pump, and this requires more mitochondria sitting on the nodes Ranvier. Okay.
Thank you. Next question from Haiyan. Are there ways to pharmacologically block synaptophyllin function?
Yes, and we are looking for some compound to. This is another directions for pharmacology, and also we're working on the bioenergetic compound as well to enhance the mitochondrial transport and also sort of signal transduction pathways. Okay.
Another question from Jiming is the latent herpes simple virus in the neurons. Is it possible to use mitochondria to migrate to peripheral nerve during reactivation? Very interesting question.
Yeah, very interesting, and we haven't thought about this yet. Yes. Yeah.
It's like a little bus.
Mm.
Another question from Mihong, is SERT selectively packed into exosome?
Yes, it means so to yes.
The oligodendrocyte story, I guess.
Yeah, oligodendrocyte cell. So too is the not expressed in neuron, very low level. We cannot detect. Okay, it's but highly expressed in oligodendrocyte cell, and based on the structure, oligodendrocyte cell and the axon, okay, that's more close to the axons. The first delivery is a signal intracellular molecule delivery for exosome to the axon first. So axon accept and so to first, then delivery to translocate to the mitochondria in axon terminal.
Okay, so 10:30, perfect timing. Thank you.